morning, or whatever time it is when you're watching this video. For me, it is currently about 7:25. I'm going to repeat myself as I've done in all the other videos, just in case this is the first video you're watching. The procedures are, throughout this lecture, I'm going to give a series of attendance words or phrases. I'm going to say, hey, the next word for attendance is, then I'll put it out there. When I say that, you need to write it down. You also need to take a screenshot of when I said it. At the end of the video, you want to email me a list of those words along with the accompanying screenshots. And just as a heads up, the last word for attendance is going to be me asking you to ask me one question about the material covered in this lecture. Even if you think you understood everything, it just demonstrates, at the very least, it demonstrates that you paid attention. But also, maybe perhaps there is something you didn't understand, or more than likely, what has been the case recently is you're asking questions not necessarily exactly about what I lectured on, but tangent to. So, for example, in a previous lecture recently, we discussed lactose, and then someone asked, why are people lactose intolerant? So it's not important for the semester, but it was relevant, it was related, so that sufficed. Anyway, that's it. The first word for attendance is kangaroo, like the bouncing, jumping mammal / marsupial from Australia. Kangaroo is the first word for attendance. And that being said, you can go ahead and skip ahead about 35 minutes, 34 minutes into the video. At that point, we should be starting the lecture. Actually, maybe about 30 minutes, because I'm going to start the lecture a little bit early today with the announcements and reminders for the students who are here early. That way we can get more actual lecture done during lecture time. But yeah, see you then. Morning. Uh, Here it's paid on 1.5. Yeah. But it's syndicated out of Q95 and put it off. The these are the ones that are over, like when we were overseas, they're the ones that were played on the encore network. So I listened to it. That's true. Same finish watching would be good. Obviously, reading would be better, but if you're watching stuff, watching is better for your brain. That makes sense. You take everything like so short, clips, the reels, too short. Memes. So I guess that makes sense. It'd be beneficial for our brain to take large stories.
I suffer through huh?
So that's things. So I feel, that's kind of how I am now. I didn't used to be that way. And, uh, really good in undergrad because I could easily just memorize crap. And a lot of the classes I took just required memorization and regurgitation instead of like, here's what you learned. Now use what you learned to problem solve. And unfortunately, this class is a lot like that too, but it's a 100 level class, so it's what you get for a 100 level class. I don't want to be too challenging. All right, let's see. Why is that not working? Interesting.
People are crazy.
I concur. What prompted that?
Huh?
I said, what prompted that this time, though?
Well, if you want it insist, I can show you.
Sure. Oh my
Yeah, there you
God.
go.
Wow. You know, that's almost a silly, stupid prank, but I bet the guy gets have to register as a sex offender for that. I bet. I know a guy who got caught. peeing outside, so, the wrong person saw him. And to register as a sex offender.
For peeing outside.
Yep, because, it wasn't written like that, it was written like exposing himself to the public or something like that.
Oh, you mean, public indecency or.
Yeah, something. Tell a tall person did this. No. Oh yeah, makes sense. Substances. Are. Yeah. Good morning.
Good morning.
Morning. Excuse me. All right, so Hill was here first. Get a lecture credit for that. Stover is here and WBSU here. Extra credit for that. Zweiback is here. Is that a WBSU shirt or just a regular old shirt? Regular shirt, okay. Champion came in, but I don't remember if he's wearing a WBSU shirt or not. I think he is. Morning. Slaughter is here. And it's WBSU here. Champion. Hey! It's gonna do it that quickly. Thanks.
Yeah, it's a second walk.
Why can't they just heathens on
Yeah.
the computer?
Send somebody at 7:49 a.m. Do not have 7:49. Or stay up, spend the night. Jeez.
You guys don't have a night crew?
I don't have a night.
That's too. Trying
You only
to,
get one though, there's another one going to see.
that's fine.
Okay.
Here's some good advice for you guys. That was just inspired by one of my other students. When you guys email your professor, it helps to say which course you're in. Some professors have a lot of courses. So, you might say, hey, this is so-and-so from sociology class at 3 p.m. Just wanted to let you know, blah, 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 blah. And obviously, you can look it up, but sometimes that just takes more time.
I don't even know our section.
Huh?
I don't know our section.
You should. That's good. I'm glad you said that. 03, 101-03. Oh, yeah. For you guys, speaking of which, this is also good advice specifically for this course. Whenever you do those extra credit things or the videos, like, you have to do a video for this week's pre-lab. When it asks your section number, put your section number. Because if I can't read your screenshot, I'm going to have to look it up. I'm going to search for people who are on 101-03. If I can't read your screenshot, and then I search for people in 101-03, and I don't see you, then you'll get a 0 for that, right? Because I need to be able to find you. So make sure you put 101-03. Of course, if the screenshot, if I can read the screenshot, that won't matter, but these are all fail-safe mechanisms, right? You got the screenshot, so I don't have to look it up. And then if I do have to look it up, you've got your 101-03. So I'm so glad you asked that. All right, let's see. All right, let's do some attendance. Brumfeld is here. Not wearing WVSU gear though. Just checking. Oh yeah, Brumfeld's there, usually there, right? Yeah, sorry. Tompkins.
She woke up.
Wait, yeah.
Oh, you are? Okay. Okay. I'll get back to you. Gorby is not here yet. Gross is not here yet. Gunther is here. You wearing WVSU gear right there? Yep. We'll let you credit for that. Randall Hill is here not wearing WVSU gear. Other Mr. Hill, are you wearing WVSU gear?
I forgot to.
That's okay. It's extra credit, won't hurt your grade, it just won't help it. Jackson is not here. Keys, not here yet. Perez is not here yet. Sigmund, not here yet. Slaughter is here. Smith is not here yet. Stover is here in WVSU gear. Tompkins is here in WVSU gear. Vanessa is here. Watson is here. Young is not here yet. Zweiback is here. Lawrence, not here yet. Carter, not here yet. So I really want to get through a lot of this lecture today, so I'm going to go ahead and start the announcements early, because some of them are just really repetitive, and there's no need for me to waste lecture time repeating it. For example, today, the lab is at 9 a.m. Make sure you're here on time. Make sure you have closed-toed shoes and pants on. It's the same stuff as usual. Any questions about lab today? Okay. I'll wait till everyone else gets here for that. So pre-lab, again, like I mentioned on Monday, that's due this Friday. This particular pre-lab starts with a video and a set of questions. So do that first, then read the introduction, then answer the questions to prove that you read the introduction, which is a good time for me to remind you the questions that come after the introduction, they provide proof that you read it. So whatever you do, don't look up those answers somewhere else. Because it's not just that I want you to know like the definition of diffusion, for example. It's that I want you to know, I want to make sure that you actually read the introduction. So keep that in mind when you're doing the pre-lab questions. It's not just answering the questions, it's also demonstrating that you read the introduction. And then you'll have 4 sets of hypothesis questions after that. They're all highlighted in green. So should be pretty self-explanatory. But as always, if you need help, let me know. because these pre-labs are not a quiz, they're not an exam. You don't have to do them on your own. I'm more than happy to meet you online and do them with you. Let's do some updating here. Perez is now here. And Young? Yep, and Young. Have I missed anybody else so far?
Exam due Sunday.
Huh?
Exam due Sunday.
Yep, exams due Sunday, like I said, on Monday, and like I'll say again on Friday. I'm curious to see who's the person who's gonna miss the exam, despite me reminding everybody at least 3 times verbally, and of course, you're gonna get some reminders on the announcements too. But yes, exam 1 is due on Sunday. It covers chapters 1 and 2. We're finishing up chapter 2 today, hopefully. I'm gonna go quick, so if you need me to slow down, let me know. It takes 50 minutes, or you have 50 minutes to answer 50 multiple choice questions because it's closed book. The idea there is for you to not have time to cheat and look up the answers. So if you want more time than 50 minutes, if you can meet me online, we can essentially eliminate the time limit if I can be sure that you are not looking up your answers you're taking a closed book exam. All right, it's 8 o'clock. No one's officially late until 8:01. So another thing I want to remind you is that I'm here for you. I had a meeting yesterday with alumni and retirees and staff and faculty, and I just wanted me to remind you that I really am here for you, not just for this class either. Like, I want your experience at WVSU to be great. So if you have problems, even if they don't involve my class, or if you have ideas of how your 4 years, hopefully 4 years at WVSU can be better, let me know. Like, I am here for you. So even, again, so if something's out of my wheelhouse, that doesn't matter. Like, I might know who to go to. You might have an idea of, like, I wish they did this for the parking lot. Like, all right, well, I'll let the right people know. All right, back to attendance, because it is 8, and by the time we finish this, no, it's 8:01. So now we'll run through attendance one more time. Since we're recording, I can go back to the video. Brumfeld is here. Champion is here. Gorby is not here. Gross is not here. Gunther is here. Both Hills are here. Jackson is not here. Keys is here. Perez is here. Sigmund is not here. Slaughter is here. Smith is here. And WSU here? Yeah, perfect. All right. I want you credit there. Stover is here. Tompkins is here. Vanessa is here. Watson is here. Young is here. Zweiback is here. Lawrence is not here. And Wilson is not here. All right, let's jump into it. Like I said, I want to go quickly because you have an exam on this on Monday. So the quicker I get it, by Sunday. So the quicker I can get this done, the more time you'll have to study it. Also, we're going to have a lab on a lot of this material today. So, first word for attendance is going to be buns, like hot cross buns. Because for some reason today is National Hot Cross Buns Day. So, buns is the first word for attendance. As always, you guys don't have to turn them in, but if you do, make sure you do it before 9 a.m. to get your extra credit. And if you're online, make sure you do the screenshots. So, before I move on, does anybody else have announcements or reminders that you want to give us? Or any questions about what's up on here? Oh yeah, the vote, sorry. Just so I can do things better next semester. Won't help you guys, but it'll help us, students after you. Historically, when we do that little pH experiment, it's been me and the TA, and we do it, and everybody watches and pays attention, and then we put the data on the board, and that's it. What you guys did was broke you down into 4 groups, obviously, so you had a bunch of people, working on that. And then, well, the other thing we could do is have 2 students per group, so every 2 people would do their own experiment. So by show of hands, how many, if you could choose, would do rather just one experiment and everybody uses that data? Okay, how many would prefer to do what we did? Okay, all right, I'm not gonna count just by looking. And how many of you would prefer 2 students per little pH experiment? Okay, so it seems like most of you just don't care, maybe, which is all right. All right, thank you for that. Anyway, once again, to remind you, we are talking about biological molecules. There's 4 big categories of them. There's carbohydrates, which we have finished. Now we're into lipids. And under lipids, there's different categories. One of the ones we talked about, finished talking about, was triglycerides. We're done talking about that, aka fats. Then we talked about steroids. We're done talking about that. The last one I'm going to mention very quickly, only because it's in the textbook. I'm not going to test you on it, and for the rest of this semester, it's an unimportant concept. But wax, right? Wax is a hydrocarbon chain that has an alcohol group at the end and a fatty acid. So not that it matters, because again, I'm not going to test you on it. But a hydrocarbon chain, which you should think of, even though I don't think I've used that term before, when we were talking about fatty acids, remember I was showing you the lipid had the glycerol molecule and the 3 fatty acids sticking off of it. Those fatty acids, that's basically the hydrocarbon chain. But again, it just at the end of it, it happens to have an alcohol group, which is OH. You don't need to know that. It's in your textbook. That's the only reason I'm mentioning it. Where can you find it? As far as animals are concerned, you can find it in beeswax and lanolin. And in plants, they use it to protect their leaves. That's basically it. So again, as far as the exam is concerned, and the rest of the semester is concerned, this is unimportant. We'll never talk about it again. Any questions about that? All right, here's the next one we're going to talk about. So again, we're talking about biological molecules, 4 different categories. Carbohydrates we're done with. Lipids we just finished. So now we're going to go into the 3rd category, which is proteins. This is a very important category. We're going to be dealing with this for a lot of the semester. You really need to understand how proteins work to understand how this other stuff I'm going to teach you works. So proteins, first of all, they're very diverse, which will not be a test question, just so you know that. You do need to know that second bullet point, though. Proteins are polymers of amino acid monomers. So that is the monomer. Amino acid, those are the monomers of proteins, and you need to know that. Because remember, I told you a lot of this exam, when we're dealing with these biological molecules, it's going to be able to differentiate between the 4 types. And one of the ways you're going to do that is knowing their monomers and their polymers. So I'm telling you now, the monomers of proteins are amino acids. I'll tell you the polymer later. Of course, the monomers of carbohydrates, just to remind you, which we already talked about, are monosaccharides. And technically, lipids don't have monomers, right? Because they're not technically polymers. But yes, you do need to remember that triglycerides are made up of that glycerol molecule with 3 fatty acids. You do need to remember that the steroid is 4 rings put together. But yeah, that's the monomer. So now that you need to know this, well, you probably don't need to know this. I'm going to put a question mark. But remember I told you, actually, this is a good question. Let's see who knows this, because you need to know it for the exam. So our cells, how much water is in our cells? There's a range. It was blank to blank percent. Anybody remember? Yes, close enough, right? If you remember that, then I'm not going to be so picky on the exam, this multiple choice exam, that would work. 70 to 95% is how much water our cells are. You need to know that. Anyway, the point is it's a lot of water, right? So then the question is, well, what happens if you remove the water? Well, if you remove all that water, over half of the weight of your cells is protein. So that should tell you something, right? Nothing's on accident in biology. That means proteins are very important, because when you get rid of the water, your cells are mostly proteins. They're instrumental in almost everything cells do, which is why I'm saying this is so important, because once we're done with this chapter, we're still going to come back to proteins. You really need to understand how proteins work, and they are the most sophisticated molecule in the human body. So, again, as far as the exam is concerned, these are the 2 bullet points you should concern yourself with. Especially this one. This one will definitely be on there. You need to know that the monomers are amino acids. And you might need to know that the majority of your cells, other than water, are made up of proteins. So any questions so far? All right, we're going to talk more about them. Here's some examples from the old textbook. You do not need to memorize this, but just so you know, there's different types of proteins. There's structural proteins, which is not going to be very important in this semester. We might talk about them every once in a while. There's storage proteins, like things that you would have in seeds and eggs. Those are actually important. That'll come back later. They provide, we'll come back to it. There's contractile proteins, which are very important in your everyday life, because that's like your muscles. That's how you move. That's how your heart pumps, things like that. But as far as the exam is concerned and this semester is concerned, that's actually not very important. We're not going to talk about them very often. This one's very important. These last 2 are very important for this exam and for the semester, well, more so for the semester. But this last 2 categories, there's transport proteins. So you're going to learn about this later, how things come in and out of your cell, because not everything can go in and out of the cell membrane. Sometimes we need proteins to help do the job. And then there's these things called enzymes. And you're going to learn about those here today and then later in the semester. So these last two are very important. You're gonna learn about them. But again, there's not gonna be a test question where it says, what are the major types of proteins? So don't worry about that. This is more of a preview of what you're gonna learn about. Here's something you need to know. Proteins have a common set of 20 amino acids. So to back up, to remind you, I just told you previously, a couple slides ago, the monomer of proteins are amino acids, and that's something you definitely need to know. And now I'm telling you there's 20 different types of them, and you need to know the fact that there's 20 different types of them. Might be on this exam, it'll definitely be on a later exam, because we're gonna dive deeper into enzymes and proteins a little bit later. But yes, you need to know that. There are 20 different types of amino acids. And we skirted around that fact when we were talking about carbohydrates, right? So the monomer of carbohydrates are monosaccharides, right? And we gave you some examples like glucose and galactose and fructose, right? So we gave you some examples, but I never gave you a number. I didn't tell you exactly how many there are, and you don't need to know it. You can look it up for extra credit if you want. But this you need to know. There are 20 different amino acids. That will be important later in the semester. So for the first time too, now we're going to look at the structure of a monomer, right? When we looked a little bit, I showed you some pictures of some glucose, some monosaccharides, but we didn't really talk about the structure of it, and that wasn't important. In this case, it is. So you need to know the structure of an amino acid, especially later in the semester. But for now, this is just an introduction. So an amino acid always has a carbon in the middle. And as you should know by now, because of the lab and because you're about to be tested on it, the carbon bonds 4 times, right? So what does this central carbon bond to? Well, there's an amino group, a hydrogen, a carboxyl group, and an R group. What you don't need to know is, like, I'm not going to show you a picture and be like, all right, take away these words and say, which one's the carboxyl group and which one's the amino group? You don't need to be able to identify it. As a matter of fact, most likely I'm not even going to ask you about the amino group and the carboxyl group. But you definitely need to know that these 20 amino acids, each one of them, any version of them, they all have that carbon in the middle, and then the bond, they've got these 4 parts. One part you definitely need to know, though, and that's this right here, the side chain. The side chain is very important because that's the only part that's different. So for each of those 20 different types of amino acids, this is all the same. The same carbon, the same amino group, the same hydrogen, the same carboxyl group. The thing that defines the amino acid is the side chain. So that is the definition, right? That's what makes whichever one of those 20 amino acids, that's what makes it. So make sure you know that. You could basically say the side chain defines the amino acid. Or if you want to turn that into one big sentence, there's 20 different amino acids, 20 different versions of these amino acids, and they're all the same except the side chain. Each one of them has a different side chain. So any questions so far? Am I going too fast? Okay. So here's everything I just said, but in writing. So again, there's 3 groups that are common to all amino acids. They all have that central carbon attached to a carboxyl group, also attached to an amino group, also attached to a hydrogen. Also, just so you know, not that you need to, but when these monomers come together in these dehydration reactions, this is actually where they get attached. The carboxyl group of one amino acid attaches to the amino group of the other amino acid. I'm not going to test you on that, I'm just letting you know. So the variable component, right? So again, all parts are the same. The variable component is the R group. The R group defines the amino acid, right? That's the only part that's different. So again, I'm going to move past this because we've already talked about this, but now you can see it in writing. Here's yet another picture, right? This looks slightly different than the other picture I showed you because this is from a different source, but it's the same information, right? Central carbon attached to an amino group, attached to a hydrogen, attached to a carboxyl group, and then finally that defining R group. So, any pictures so far, or any questions so far? Again, I'm beating a dead horse here. Hopefully you'll get it. Each amino acid has a unique side chain. So, right, that part I've said it a few times, and hopefully you got it by now. But what I haven't mentioned yet, that hopefully is kind of self-evident, is that these particular side chains give it its special chemical properties. So each one of these 20 amino acids is slightly different from each other. They have different chemical properties because of these side chains. So again, it's the side chain that defines which amino acid it is. Not only that, but again, it gives it its special chemical properties. The next slide I'm going to show you shows like a really generic version of two different real amino acids. Again, when I show you this, keep in mind, I know that you're not chemists, I know that you're not biologists, but just remember what I've said earlier about like form and function. And at the molecular level, all these things fit together in different ways. So let's take a look. And these are two basic amino acids. A lot of them are a lot more complicated than this. But here's leucine on the left and serine on the right. These are examples. You don't need to memorize these. But you can see here, this has this shape, right? This has a smaller square shape. And without being a chemist, you can just tell by common sense that these things are going to fit into things differently, especially when it comes to proteins. When it comes to proteins, I don't think I've said this yet. Just so you know, proteins are very much about the shape. It's very much about how this thing fits into that thing, like a lock and a key. And we'll talk more about that later. But the shape is very important for a protein. So even though you may know nothing about chemistry, you just look at the size and the shape of those two things and realize they fit into different holes, so to speak, right? They fit into things differently. And that's just the size and the shape. Then we do throw a little bit of chemistry in there because you do know what hydrophobic means, right? Hydrophobic would be like oil, lipids, things that are attracted to each other but are repelled from water and repelled from other hydrophilic things. Here we have something that's hydrophilic. So these two things will repel each other, right? This thing would be repelled from water. This thing would be attracted to water. This thing would be repelled by anything that has a charge or a partial charge. This thing would be attracted to it. So again, there's 20 different versions of these, right? And they all have a different size and shape with its own unique chemistry, and that dictates how they fit together. And again, with proteins, it's like the way they fit together, it's more important than any of the other molecules we talk about. So here we go. I've already told you, and I mentioned it today, and see if you can remember, because you need to know for the exam, and I just said it, probably less than 5 minutes ago. When you bring two monomers together, what kind of reaction is that? And it produces, it takes water out of the reactants and gives us water in the products. So what kind of reaction is that brings two monomers together? Hopefully you're just being shy because you're going to be tested on it by Sunday. That is a dehydration reaction. You need to know that. But just to make things even more complicated, biologists were like, let's give this one a name too. So technically, when we're bringing two amino acids together, it's called a peptide bond. I'm almost definitely not going to ask you that. And the only reason I even bring it up, well, first of all, is because it's in your textbook. But also, the name, remember, you already know the monomers. The monomers of proteins are called amino acids. We just got done talking about them. So now I'm giving you the name of the polymer because you'll need to know this for the exam. The name of the polymer is called polypeptides. So you definitely need to know that second bullet point. And because you definitely need to know the second bullet point, that's the reason I brought up this first bullet point. Just so you know where they got the name of it. Where the hell did polypeptides come in? Why not call them polyamino acids? That's what I would call it. But yeah, they're called polypeptides because technically those dehydration reactions, they create peptide bonds. And just so you know, a peptide bond, it's just like any old covalent bond that you've already learned. But someone just decided to give them their own little name. But yes, so again, to back up really quickly, you need to know that proteins are made up of, the monomers are amino acids, and the polymer is called a polypeptide. Then it gets a little bit trickier with this second main bullet point here. A functional protein is at least one polypeptide that has assumed a certain shape. So this is where it gets trickier. Let's see here. I'll give you an example. Yep. What is this that I'm holding? Correct. That's a piece of paper. Now, bear with me. I should have done this before the lecture started. A good at this in elementary school. Now what am I holding? Paper airplane, but she just said it's a piece of paper. You saying she's a liar? No. It's still a piece of paper, right? But it's also, we can now consider it a paper airplane because it's been folded into this particular shape, and now it can do its job, maybe, which is to fly. No. Fifth grade me would be so angry at old man me. I used to be able to make it fly. But anyway, once it's folded into its shape, it is now considered a paper airplane. And it can do what it's supposed to do if it was folded properly. This is the same relationship between polypeptides and proteins. So a protein is a protein only because it's shaped and twisted and folded into its correct shape. It's all about the shape. If it's not into its particular shape that it's supposed to be into, at that point, it's still just a polypeptide. So a polypeptide is a string of amino acids, right? That's the polymer. But then once it gets folded into the shape it's supposed to be in, we then consider it a protein. So make sure you understand that relationship, not only for the exam, but for later in the semester. Because again, proteins are very important for the stuff we're gonna learn. And this is why I have this picture of, it's kind of hard to see, but that's like a crocheted blanket, right? So if you have a piece of yarn, it's just a piece of yarn. But then when you crochet it into its particular shape, it's still a piece of yarn, but you now consider it a blanket or a scarf or whatever it is that you crochet. So any questions about this slide? All right, the next word for attendance then is going to be yarn. Since we have a bunch of blue yarn there, I don't know what it makes up, but I think it's, like I said, I think it's a blanket. So if there's no questions, here's a picture of something I've already mentioned. Here's two amino acids coming together. Like I said, not that you need to know this, it's always the carboxyl group from one coming together with an amino group on the other. What you do need to know is that these things are dehydration reactions, right? When you bring things together, as far as we're concerned in this class, it's always a dehydration reaction. Also to remind you, even though it's not on the screen, if there was a chemical formula showing you this, you would see water on the product side, right? Because we're producing water, that's what makes it a dehydration reaction. We're taking water out of the reactants and we're getting it in the products. And yes, technically that chemical bond between the carboxyl group of one and the amino group of the other, technically that covalent bond is called a peptide bond, but again, it doesn't matter for us. I just included it because your textbook did it and to remind you, hopefully help you remember that these are called polypeptides. Excuse me. So, here's something interesting to think about. Your body has thousands of different types of proteins. I don't mean like thousands of proteins, like your cell itself has probably millions. Each one cell probably has millions of proteins in it. But I mean different types of proteins, right? So there's thousands of different types of proteins in your body, yet you only have 20 amino acids. So how is that possible? Not that you're going to get tested on this, but I like to compare it to the English language, right? So the English language only has 26 letters, but if you put them together in different combinations, right, then you can make all these different words. And in a sense, it's kind of the same thing. And that analogy is going to come back much later in the semester. So you have these 20 different amino acids, and depending on how you put them together, it means a different thing. It does a different thing. Same thing with the English language. Of course, there's more rules for the English language about, these letters always go with this one, and these always follow those. But last time I checked, and I don't know what it is now, it's probably a lot higher than this. So with 26 letters, we have more than a million words from 26 letters. So that's how we can have thousands of proteins in our body, but only have 20 different amino acids. It's all about how we put them together. If you download the PowerPoint, you can watch this little animation that talks about something called primary structure, and I'll get into that in a second. But just so you know, primary structure, like what I just described, when I said, all right, this amino acid goes with this amino acid, goes with this amino acid. When you're building that polypeptide and you're only concerned with which amino acid goes with which, that's called primary structure. And again, if you download the PowerPoint, you can watch that. So that being said, it's the sequence of those amino acids, like this one, this particular one is attached to this particular one, which is attached to this particular one. That sequence actually determines its shape. So it's not consequential, right? Because as I mentioned earlier, when we were talking about those R groups, which defines the amino acid, they're all different. They fit into different things. Some of them are charged, some of them are not charged, some of them are hydrophobic, some of them are hydrophilic. So they all fit together in different ways. So the sequence of amino acids, right, how you put those 20 different amino acids together determines how it's going to fold upon itself. And this is a great example. Not that you need to memorize this or even completely understand what this is, but here are these two things. These two amino acids both have this little sulfur sticking off of them as their R group, and because of that, they're attracted to each other. So now, instead of having a long, straight strand of amino acid, this thing is folded up on itself because these two are attracted to each other, right? And that's just one example. Imagine thousands, millions of these interacting together. But again, just because this thing happened to be here and this one happened to be here, that caused that thing to have a kink right there, right? That caused it to fold up on itself. So that's how this works, right? Each amino acid has its specific R group, which has its own specific chemistry. Because of that, that determines the three-dimensional shape. And again, it's all about the shape when it comes to proteins. It's very important, not just for this exam, but for understanding things later in the semester. Any questions? Okay, here's a picture. I think this one actually is from your textbook, which is good because probably it's hard for you to read this. But if you look at it in your textbook, you can see it and make sense. But this is going back to what I was saying earlier. Primary structure, and I'm probably not going to test you on this, but when someone's describing a protein and they're talking about the primary structure, they're literally just saying which amino acid is attached to which. Well, first it's a leucine, then it's a serine, then it's a tryptophan, whatever it may be. Primary structure is that's all that it's about, right? Talking about which is attached to which. Secondary protein structure is the hydrogen bonding of the peptide, blah, blah, blah. So it's the thing I showed you earlier, right? So some of these things are attracted to each other and that causes it to kink in or maybe repel each other, whatever. So that's secondary protein structure. Tertiary protein structure is the three-dimensional folding. You don't need to worry about that. Quaternary protein structure, sometimes you have multiple multiple polypeptides coming together to form one protein. All this, as far as I'm concerned, from here down, right, that's all above 100 level stuff. So you can read it if you want, it won't hurt you. But for now, especially at the beginning of the semester, we're really just focused on that primary protein structure, which is knowing this amino acid is attached to that amino acid, and that dictates ultimately the shape of the protein. So here's one example. Again, just an example. You don't need to memorize this. This is from the old textbook. They had to curl this thing around just to make it fit on the screen. But there's nothing, as far as bonds are concerned, that are making those curl. That's just the way they drew it, right? But when you actually put them together, they do fold up in certain ways, and this is what it looks like. It's just an example. You don't even need to know what that means. Again, you can just look at it and be like, okay, that thing has a particular shape. It has a particular function. That's all you need to know. And of course, different proteins would have a different shape. Therefore, they would have a different function. Matter of fact, there's probably some other protein that has a shape that fits just like this because it would fit in with that protein beautifully, right? And that's a lot of times how proteins work. It's how they fit in together like puzzle pieces, like three-dimensional puzzle pieces. Anyway, that's just an example, so we'll move forward. Let me know if I'm going too fast. I'm trying to get through this. So obviously, if I'm telling you it's this sequence of amino acids that dictates the shape, then hopefully you can figure this out without me telling you. Hopefully I could ask this and you could have answered it, but I'm moving quickly, so I'm not going to waste time doing that. But if you change the sequence, that obviously can change the shape. So going back to that other picture I showed you where it was like, I think it was shaped like this, basically, because you had two of them that were attached to each other, like bonded to each other. Because of that, because you had that one particular amino acid here and that one particular amino acid there, it caused that protein, that polypeptide to fold in on itself, right? So if you were to get rid of that, if you were to change just that one sequence, then there would be no reason for that thing to curl in like that and fold in like that. So then you would just have another straight polypeptide using that very basic example. So again, just you changing that one amino acid would have changed its entire shape of the protein. And again, protein, when it comes to proteins, the shape is the most important function, or the most important thing about it. If you change the shape, you change its function. And again, if you change one of the amino acids, just one of them, you're probably going to change the shape. Therefore, you're probably going to change the function. You don't need to know this yet. We're going to bring it up a few times in the semester. So these are just examples. I don't think I'm going to ask you about it. Definitely, maybe later. Definitely not right away. Not in this exam. But the example they give is hemoglobin, right? So if you change just one protein in this protein, one amino acid in this protein that's in your blood cells, then that changes the shape of it. And when you change the shape of it, you change its function. And that causes sickle cell disease. Has anybody heard of sickle cell disease? Yeah. Well, it's caused by just that one amino acid being changed. And that will be important later in the semester. We're going to use that when we talk about mutations. We're going to use that. We're going to talk about that again when we talk about natural selection. So this example of sickle cell, it's going to come back. But anyway, yes, one change, the amino acid can change the shape. And if you change the shape, you change the function. So it's a pretty big deal. So again, here's an actual picture of what it is. This is the one change. I forget 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So from I don't know, I drew it wrong. Anyway, they changed one amino acid, right? You have one amino acid that's different of all these amino acids. You just have this one amino acid that's different. And now instead of having this, you have this, and that it might be hard for you to see the difference in that. But then if you look at what the blood cells look like, that's a normal red blood cell, and that's a sickle cell red blood cell. And that causes, again, sickle cell disease, all because you changed one amino acid. So again, everything I just said, here it is in writing. One difference is enough to cause a protein to fold into a different shape, which then would give it a different function, and very often that can cause diseases. Of course, as you're going to learn much later in the semester, sometimes it's good. This is how we get new proteins. Sometimes we have these mistakes, and then we get a new protein with a completely new function that can actually sometimes be beneficial. Because of that, we have natural selection, but we'll talk about that later. Anyway, later too, we'll also talk about prions and other severe nervous system disorders, but for now, that's just the introduction. Once again, if you change one amino acid, much less a bunch of them, then you're going to change the shape. When you change the shape, you change the function. Here's an example. Again, we'll talk about prions later in the semester. I'm not going to ask you anything about this on the first exam. But in your brain, you have these normal proteins with their normal shape. Prion is a misfolded protein. Not only is it misfolded, but it's also kind of contagious. So when it comes in contact with other proteins, then they become misfolded. And eventually they cluster up and they have then you get big holes in your brain, which is where you get that word encephalopathy. It's always a hard one for me to say, which basically means sponge brain, right? So because you get these holes in your brain. And yeah, I'm sure you guys have heard of mad cow disease, which is that example. If you guys hunt, you probably heard of fatal weight loss in deer or elk and stuff like that. So that's real stuff. And Kuru, that's an interesting one if you want to look it up for independent work. Kuru is spread by cannibalism in humans. Because there's certain people in certain parts of the world where they would, I don't think they still do, but they would eat the brains of the people that they beat in combat. Anyway, they're eating the brains, and then they get it, and they would die from it. Anyway, any questions about this? Just an example. There's going to be nothing on the exam. All right. So again, we're keep talking about how protein shape are so important. So let's talk about this too. Not only would changing the amino acid change the shape of it, but the shape is also sensitive to the environment. This is important, not just because you need to know this, but also this is a good throwback to some of the stuff that you learned earlier. So a change in a temperature, for example, could change the shape. Because remember, temperature, as far as we're concerned in this class, that's all about molecular movement. So if we're saying high temperature, we're just meaning molecules are moving faster. Low temperature means they're moving less. So when you're increasing the temperature for this protein, right, you're adding energy to it. And hopefully you remember this, you need to for the exam. You can see these hydrogen bonds here, keeping this together. Hydrogen bonds, as you need to know, are the weakest of the bonds. So there's energy keeping that together, but not much, because this is the very weak bonds. So if you add some temperature to it, you're going to break those bonds. If you have enough temperature, if you add enough temperature, those bonds are going to break, and that thing's going to unravel. It's going to lose its shape. It's going to lose its function. Same with pH. So for example, here's something else you need to know for the exam. This is why I'm saying this is such a good throwback slide, because it's a good review for things you need to know for the exam. Oxygen, so if we're talking about hydrogen bonds, the oxygen ends are slightly negative, right? Because that's where all the electrons are hanging out. the hydrogen end is slightly positive, right? So, and here's another thing you can throw back, something you need to know for the example. When we talk about the change in pH, when we say the pH is going down, what does that mean on a molecular level? pH is going down means you have an increase of what in the solution? And I give you the choices. Alphabetically, we have H pluses or OH minus. So when we say pH is going down, what does that mean? We're getting an increase in what? The H plus or the O minus? Hopefully you know, and you're just too shy. I don't have time to wait for it because I'm trying to get through here. But just to remind you, when the pH is going down, that's an increase in H plus. When the pH is going up, that's an increase in OH minus. You have to know that for the exam. It's not just trivial. It's going to be important later in the semester. Anyway, so if we say, for example, that the pH is going down, right? You've got this protein, pH is going down. That means there's going to be more of these H plus molecules, right? Well, not molecules, these H plus. We won't say molecules. But here we have these hydrogen bonds. And remember, hydrogen bonds are between partially charged things, right? That's a partially negative oxygen. That's a partially positive hydrogen. So this partially negative oxygen is going to be attracted to that fully positive hydrogen, right? It's not going to be attracted to this partially positive hydrogen anymore. It's going to be attracted to that fully positive hydrogen. Therefore, all these hydrogen bonds are going to break. Therefore, that thing's going to unravel. And therefore, it's not going to be the same shape. It's not going to do its job. The opposite would be true if you raise the pH too much, right? So here we have these OH minuses. This hydrogen would be attracted to that fully negative OH minus, right? It would be no longer attracted to that partially negative oxygen. So that's how a change in pH works. Long story short, I know I went into that a lot because it's a lot of review that you need to know for the exam. But as far as the exam is concerned and this slide is concerned, protein shape is sensitive to the environment. That's what you need to know. I'm not going to go into that much detail about temperature and pH, but again, or at least not when I'm talking about proteins. But you do need to know all that stuff. You do need to know H plus, OH minus, hydrogen bonds, partially negative, partially positive. It's a great review slide. Woo! We got through proteins. Let's see if we can get through the last one, which is nucleic